Hello and welcome to IB Bio 1.6 notes. So first off, we're going to talk about the cell cycle. So there are three stages to the cell cycle. We have interphase, mitosis, and cytokinesis. So interphase is the longest part of the cell cycle, and it also includes three parts. So we have G1 right here, S, and G2. So it includes common cell processes such as metabolism, endocytosis, exocytosis, and using and obtaining nutrients. So G1 occurs in the cytoplasm, and it's when the cell grows and functions normally, undergoing everyday processes. However, rapid protein synthesis takes place, allowing the cell to grow in size. But the proteins required for DNA synthesis are made, and the mitochondria and the chloroplasts are replicated, which also continues in the S phase. So in the S phase, this takes place in the nucleus. DNA replication takes place, and the genetic material is duplicated, but no chromosomes are formed yet. And then we have G2, so this occurs in the cytoplasm. Protein synthesis occurs to produce the proteins needed for cell division, and the cell is actively preparing for cell division. Then we have mitosis, which is the second part. So it's the division of the nucleus into two genetically identical daughter nuclei, and it involves separation of sister chromatids into individual chromosomes, which are then distributed among the daughter nuclei. And finally, cytokinesis is when the cytoplasm of a parental cell is divided between the two daughter cells following mitosis. Then we have cyclins, so this is important. Cyclins are proteins that, along with cyclin-dependent kinases, regulate the cell cycle. So depending on the presence, so the concentration, and the action of these proteins, the cell cycle can be either fast or slow. So cells cannot progress to the next stage of the cell cycle unless the specific cyclin reaches a certain concentration. So for example, if cyclin E does not reach this certain concentration, then it could not move on to the S phase cycle. So cyclins bind to the enzymes called CDKs and activate them, and this activated CDK then attaches a phosphate group to other proteins in the cell, and the attachment of the phosphate triggers the other proteins to become active and carry out tasks. So this part is not really that important, just know it like in the background, specific to the phases of the cell cycle. So mitosis. There are different parts to mitosis. So first of all, eukaryotic chromosomes consist of DNA, which is tightly wound around proteins called histones. So these little proteins are called histones, the purple thing. And they're proteins that form part of nucleosomes. So we see here these are nucleosomes, DNA wrapped around histones. And these many nucleosomes coil together to form a pattern which forms a structure called the chromosome. So there are four parts of mitosis. We have prophase, metaphase, anaphase, and telophase. So in prophase, which we see on the right here, DNA coils using chromatin to condense into sister chromatids. So we have these sister chromatids right here. And the nucleolus disappears, so there's no longer a nucleolus inside the nucleus. The nuclear membrane disintegrates. So this nuclear membrane starts to disintegrate, and the spindle fibers, which you see right here, these lines, they start to form and are completely formed by the end of prophase. So you're going to need to know all these steps. And then obviously the centrioles, which are not in plant cells, only in animal cells, move to opposite poles. Then we have metaphase, which is when the spindle fibers right here, they bind to the centromere of the sister chromatids, which is the middle portion of the sister chromatids where they attach, and cause their movement towards the equatorial plate, so in the middle. And the sister chromatids are aligned at the equatorial plate by the end of metaphase. In anaphase, we have the sister chromatids are separated, so you see here they're separated. These sister chromatids are now known as chromosomes, completely separate chromosomes, and are pulled to opposite sides by the spindle fibers, which are these lines here. And during telophase, the chromosomes have reached the opposite poles. A nuclear membrane starts to reform at each single pole, and a nucleolus appears in each new nucleus, and the spindle fibers also disintegrate. And finally, the cell elongates, as we see here. It's a longer cell in preparation for cytokinesis. So cytokinesis in an animal cell is when a ring of proteins, so called microfilaments, they're located immediately beneath the plasma membrane at the equator. They pull the plasma membrane inward. So we see here this cell, it sort of pulls this plasma membrane inwards, and the inward pull produces the cleavage furrow. We see that right here. So this is the cleavage furrow. And then when the cleavage furrow reaches the center of the cells, it's pinched apart. So it just pinches on itself to form two daughter cells. You're also going to have to know this process. So cytokinesis in plant cells here, it's a little bit different. So in plant cells, the Golgi apparatus forms a vesicle that consists of material to build a new cell wall. So these vesicles merge and form the cell plate. So you see here the cell plate is forming. So these are all formed by vesicles, and the cell plate grows and divides into two daughter cells, completely separate daughter cells. Tumorigenesis. So tumorigenesis is the formation of one or more tumor, which is defined as a mass of cells that divide uncontrollably. So we have two types of tumors. We have benign tumors and malignant tumors. So benign tumors are localized and do not spread to other parts of the body, while malignant tumors are cancerous growth that is often resistant to treatment, and it may spread to other parts of the body and sometimes recur after it's been removed. So tumors obviously occur due to a mutation in one of the cyclins, which causes a disruption in the cell cycle and causes the cells to divide uncontrollably. And of course, a mutation is the change in an organism's genetic code. So mutagens are agents that cause mutations, such as carcinogens, which are chemicals that cause mutations, such as these chemicals. Also high energy radiation, such as X-rays, short wavelength ultraviolet light. So we see that a lot from the sun. It could cause skin cancer. Some viruses, such as hepatitis B. And also an oncogene is a gene that has undergone a mutation that will contribute to the future development of a tumor. Metastasis is the movement of cells from the primary tumor to other parts of the body where they develop into secondary tumors. And then some cancer cells gain the ability to penetrate the cell walls of lymph or blood vessels, and which allows them to circulate around the entire body. And these circulating cancer cells invade tissues at different locations and develop into secondary tumors around the entire body. So you're just gonna have to memorize all of this. These definitions. And then smoking and cancer. So smoking causes many cancers, one of which is lung cancer, as well as strokes and loss of diseases. So this graph displays the age of quitting smoking compared to the cumulative risk of cancer. So we have the never smoked. They obviously develop a higher risk of cancer at a larger age. Right here. A lower risk of cancer at a larger age as compared to those who have continued smoking. So this light blue color has continued smoking. They have over a 15% chance of developing cancer. The yellow stopped at 50, red stopped at 30, and obviously we see the later you stop smoking, the higher the chance of you developing a tumor or cancer. So these are just some of the many diseases that could occur because of cancer. That's it for topic 1.6. Thank you.